Thank you very much to both of our opening keynote speakers this morning. And so I'd like to move us into our first session for the day. And we have a session, I think as everyone's seen, on integrating in silico NAMs with other NAMs today. We're going to lead off with Dr. Thomas Hartung from Johns Hopkins University. And so without anything else, I'll pass it on to you right now, Thomas.
Thanks, Nick, and good morning, everyone. My wife, Lena, was supposed to be on the schedule, but we had conflicts, and I have to leave instantly after this talk to do another talk in Bethesda. So I'm very sorry. I've put what I'd always tell my students not to do, far too much material in these slides. But I make the slides available with this QR code, which is also on the last slide. Again, if you're interested in a lot of references to papers. So the big message is NAMs and AI are disruptive technologies which are coming. If you read last week's Nature paper, you see here how in the research area for 7 clinical indications, the use of NAMs-only papers in orange is rising, while the animal-only papers in light blue are more or less stable, and the combined papers are rising too. So NAMs are reality here. And you will have certainly been realizing that on the regulatory level, FDA, EPA, NIH all made major announcements over the last 12 months in moving into these technologies, which I consider a necessary adaptation to the state of art, the state of the science. And based on all of this is that there's exponential growth in many technologies which are relevant to us. This starts with analytical technologies like mass spectroscopy, which is halving its sensitivities by every 3 to 4 years, or in other words, last 30 years, we have seen a million-fold improvement in the sensitivity of mass spectroscopy. Our microphysiological systems which bring us together here are doubling publications every 4 years as an exponential growth. Genome sequencing is halving price every 1.5 to 2 years. So from $3 billion for the first genome, we are now at $200. Computers are following Moore's law. For 60 years, half the price, twice the capacity every second year. The data in the world are doubling every 18 months, or 90% have been produced in the last 3 to 4 years. AI is the fastest of all. It's doubling capacity every 3 months since deep learning was introduced. So this year's AI is 8 times more powerful than last year's. And the prices are falling. 1.5 years ago, you paid 280 times the price we are now paying per token in the AI sector. And all of this is synergistically changing things. And for toxicology and pharmacology and extension, the bottleneck was really reading. There's 3.4 million scientific papers in biomedicine produced every year. PubMed took up in 2023 2.2 million alone. And nobody can read it but AI. Toxicology, more than 20,000 papers per year. Since 2023, AI is good enough and outperforming humans in reading scientific papers if they only get access to this. And this is why I have a very strong call for open access things, and that's the basis for meaningful AI. But it's also about databases, which are increasingly becoming available. It's about the sharing of information. And these are now 2 pictures I took from a manuscript we are just submitting with Hartung from FDA. Natural language processing is really key. It is key for finding what is out. You cannot imagine how many studies have been done already and nobody finds them. It is for structured extraction of information to get this information into the computer. It is for qualifying this, for example, by risk of bias automation, finding out, is this a good source? Is it not? Because our current AI is usually very blue-eyed. They just believe what is written in the text. But if we can find out what is good information, the AI can too. And increasingly, AI can bring things together, make a mechanistic assembly and find out what is the, in our case, adverse outcome pathway. I like to call them virtual adverse outcome pathways, in contrast to the literature-driven, manually created adverse outcome pathways which we have now. And it's helping us to predict things. To show you how this toxicology, the AI-based facilitated toxicology, is moving. And this is really on the basis of big computer make big sense of big data. With the access to omics, high content imaging, high throughput data, sensor technologies, literature, internet, legacy studies curated, we really have a big database. And I would like to show you now how we come to big sense just with our own work over time. We started in 2013 to invest into AI. Our first paper was in 2015. We studied only skin sensitization. We had 1,000 data points on 150 chemicals, which we predicted, and we were 65% correct, which is not too bad. Different skin sensitization models in animals predict each other in the lower 70s. 2018 already, we predicted the 9 most common animal tests based on 600,000 data points. We predicted 190,000 chemicals for which we had data, and we were on average 87% correct. This was already outperforming the accuracy, which is on average 81% for these 9 animal tests. Last year, we were on a database of 260 million data points from just 12 databases, PubChem, Campbell, and similar, which were included. We had a holdout set of 26 million data, which were not used for predicting, and we were able to predict 4,000 properties of each and every chemical. And we're on average 91% correct for these 26 million data points. This is papers which we are at the moment just submitting. Imagine this, yeah? Whether already available, not available, it either retrieves or predicts data for millions of data points with this level of accuracy. But with agentic AI coming to us over the last, let's say, 3 to 6 months, this is at the moment exploding. Our current model has 70 billion data graphs from 2,000 databases. We are able to predict 6,000 properties of any chemical. And we are at the moment in the process of determining how much of this is accurate. But our expectations can only rise because of the law of big data in AI. This is going to be announced next Saturday. So this will be showcased the first time at the Society of Toxicology meeting, because in Silica, the spin-off company of Tom Lichtefeld, which is spearheading this work, is going to go public with this. We are talking to OECD and the Integrated Chemical Environment at NIH to host this, to donate this engine of a transformer for making it generally available. So now everything in this space is up to deployment, but the prerequisite for this is building trust. This is a paper from last April, written as last author with Robert Califf, who was at the time of writing the head of FDA, as of the 19,000 people agency at the time. And it's really about building this trust now. And there's a couple of tools here, again, from the same manuscript. It is about traceability of claims. It is about human accountability. It is about the consequent reporting of uncertainties. It's a versioning thing. Yeah, many people forget to put numbers on which version this was done with. And it is about context of use benchmarking, which cross-references to the validation paradigms which are currently developing. So I think this is really critical. What we are trying to do and what we implemented in our software, which we will release next week, is an evidence-based AI approach. Evidence-based medicine, systematic reviews with strict inclusion-exclusion criteria, defining what is the question, what is acceptable evidence, meta-analysis, quality scoring. These are essential tools which we can now put into an agentic AI. So not just take a search agent, but also have an agent which is quality controlling. And this is what we are doing here. So the best what humans can do with information is going to AI, which is on scale, allowing these type of things. So I think there's a very, very interesting time coming where these tools are being explored and implemented in various areas. So let's come to the MPS, which are bringing us together here. You know, we have been in this field quite a while with developing workshops, developing the World Summit Series in the National MPS Society. I'm putting this up mainly to alert you once again that in May we are meeting in Washington. So if you have missed to put this into your calendar, please do so now. Jay Hickman, as the president of the MPS Society, is here. For me, it is all about quality. I have been heading ECVAM, the European Center for Validation of Alternative Methods, where we published in 2005 Good Cell Culture Practice, how to do quality work similar to GLP in vivo. In a process which took us 8 years and involved many of you, more than 300 people were involved in these 8 years, we developed guidance in 2022 on good cell culture practice, including MPS. This is guidance I really ask you to take serious. At this moment, we are working on reporting standards, because it's not only to do good work in the lab, but also to report it properly. So we have published in July of last year the good in vitro reporting standards as a draft, which is under stakeholder discussion. We've received something between 100 and 200 reports, let's say comments from stakeholders, and we will in April now finalize this document. Still opportunity, we'll take up comments until the last moment. So if anybody is interested, we are interested to have more information here. In January, we published small agentic AIs to show that these reporting standards is something which the computer can test while you are writing and can say, yeah, you missed this piece of information. The GIFRES standard is not complete, but we did also publish them for the ToxR tool and for the ARRIVE guidelines for animal studies to show the principle. The publisher can now just check, is this paper complete before even sending it to an editor? And this is also, I think, the way forward for the metadata we need when sharing data for AI use, because it is saying, is this a complete description or is it not? This helps to evaluate or helps to ask the right questions. The next step, from my point of view, is ontologies, so to develop standardized vocabularies. From an upcoming paper, this is a graph just showing you there is quite a few initiatives at the moment trying to establish vocabularies in this space. And I think this is really important to make optimal use of what we are doing for the use by AI. The third step in quality control is validation. And I have a strong interest in validation. These are a number of papers which are showing where we are and where I think they could go. Some were written here with the CPAF Institute, others were lauded by NIH with an award in complementary, where we suggest, for example, to use AI more consequently in the validation process by selecting reference compounds, by emulating studies, modeling studies, by training through AI-based methodologies and others. So I invite you if you're interested in details. Lena is leading our brain organoid work, and she would have told you quite a bit about the brain organoids, which I cannot miss to touch quickly on. This is a review paper where we are describing the field and what is possible with brain organoids at the moment. What I would like to show you is that a model developed out of this with CRISPR-Cas technologies, where fluorescent reporters were cloned into these stem cells by CRISPR-Cas. Here, for example, in green oligodendrocyte lineages. This was developed with EPA funding, and we are at the moment finalizing our mini brain bow to follow non-invasively the different lines of development, and has just been chosen by the foundation of NIH as one of the 4 front-runner models now for the validation exercise. So we're very excited this is moving forward. A very interesting combination of brain organoids and AI is our work on what we call organoid intelligence. So we're trying to start producing cognitive functions in these assays, learning and memory in an organoid. And the idea is to have them play these little video games, and we can measure with these video games success in learning. And if you look into it next day, is there also memory involved? This is progressing very nicely. We are organizing the first conference in this field in October of this year in Washington. We have a community of about 250 people at the moment, which is organized through a bulletin and a website, and this is increasing. The National Science Foundation is funding, upon our proposal, now 12 projects in this field of organoid intelligence in the Begin OI program. And there's a lot of applications, both in medicine, but also for neuromorphic computing, trying to understand better why our brain is such a brilliant supercomputer, which is just 1.4 kilogram and 1 millionth of the energy consumption of a comparable computer. So this is really, I think, opening up a very interesting future scientific field. I would like to close with these disruptive technologies are coming together, and they're allowing us to do more than what we are typically thinking of, which is drug development and safety. By the integration with AI of microphysiological systems and data science, and also exposure science to some extent, we can first of all anticipate things. You need for many animal tests simply kilograms of a substance to run them. AI microphysiological systems use milligram. AI uses nothing. So we can even, before we synthesize, ask, is this benign by design with the computational tools? And this allows either green toxicology approaches as part of green chemistry, or in pharma, we call it test early, fail cheaply. And this is what these technologies are enabling. But there's also a very interesting perspective to combine this with exposure sciences. I call this exposome intelligence, and through digital twins make sense of this, what biomonitoring information, and design a human exposome as a complement to the human genome, the exposure side of disease. And the digital twins also link us to personalized medicine, where the same tools are at the moment being developed. So we have safer products, better treatment, and safer people. We held an Exposome Moonshot Forum last May in Washington, where we brought in the civil society from WHO, UNESCO, for NIH institutes to co-organizing. And we now created the Global Exposome Forum from this. I just come back from a meeting with UNESCO on Monday, where they committed to a memorandum of understanding and working together for a Global Exposome Forum. Even an African chapter is now being created by the South African government. So let me close with this. I don't have more time. I hope I've given you some ideas and sparks for following up. I think the integration of these disruptive technologies and our massive knowledge of the past, which is finally machine readable, allows to accelerate drug development, optimize public health prevention, but also future projects like green toxicology and the human exposome. Thank you very much. I will not be part of the discussion, unfortunately. Please contact me or follow here on LinkedIn.
Well, if there's one or two questions,
I'm happy to entertain any questions. Oh, there's a workout.
Is this on? Is this on? Yes, it is. It's spectacular if you can do those predictions, I think it is, with those, the number of properties, the number of compounds and so on. How do you define correct?
Correct was here the holdout data set and how well it was predicted. So 26 million of these chemicals were not used for training, or data points for chemicals were not used for training, and then they were predicted.
So what is, it's like a factor of 10 can make a huge difference in drug development, for example, right? So even a factor of 3 could.
Yeah.
So predicting properties with that precision would be impressive.
It is. No, I mean, I'm blown away. I never would have believed this is possible. But there is a lot of limitations. Yeah? For example, potency. We are only approaching this, and we have not really validated for potency. It's really a hazard prediction tool mostly. But by retrieving data, we get more no effect levels and others, and we are increasingly good in predicting GHS classes and others. But there's also tons of properties which are not just toxicological, chemico-physical properties and others. And I have spared you from the most important part, which is how we integrate these properties to mapping it on adverse outcome pathways and physiological maps to make sense. Yeah, so we're not cherry picking, but we choose, let's say, the 40 properties we know which are relevant to thyroid endocrine disruption. Yeah.
And how good does it have to be to actually make a decision based on the predictions that you're making?
From my point of view, it is about, at the moment, as a co-pilot, to put on a silver platter information you can take for your decision taking. And this will be completely context of use dependent. If you're a chemist who synthesizes and has to spend half a year to get the compound, you will be guided with something which is less clear. other cases, you will only say, oh, this flags a problem, let's request more data.
Yeah.
It will rarely be the only decision point. And we also need then the technologies to verify these predictions. We need to feed this system with experimental data, which are hopefully getting better and better.
Okay. All right. I could ask questions all morning, but
I'm happy to entertain any discussion through internet, please.
Yeah, thank you. Great presentation. And thank you for connecting the dots to the context of use. I'd love to get your additional thoughts on the trust components, which I think is absolutely important, and how you're thinking about linking that with what you just articulated beautifully about the dependency of the context of use with the decision making that the tools can inform. And so can you...
I mean, the first property of this tool is to retrieve the data. Yeah? And then if there's data gaps, to fill them, but clearly label them as predicted tools. Yeah? So the first thing is that our tool is clearly referenced. This is the database. This is the scientific publication where this number was taken. And you can later say, okay, that's a credible source or not. Yeah? Anything which is predicted is interestingly coming with a probability of being correct. Yeah? So this is AI is an inherently probabilistic tool. It is always saying this is the most probable answer. But typically, we don't ask it, and how probable is this? But this tool is trying to do exactly this. And by separating these two, clearly, you can say this is an acceptable read-across type of information or not. But it is a very young tool. But I can tell you, we have run now 12 cases where we did do a complete risk assessment of 50 to 80 pages for substances. They're at the moment under peer review by experts. The first feedbacks we got, they were blown away what these tools can collect from all of these sources on existing industrial chemicals so far. This is not for yet for the space of pharmaceuticals. And I'm looking for pharmaceutical partners and talking to several companies at the moment to tailor it more for the drug development process and how to use it there. Please. I'm happy to, it's now the moment, yeah? The tool becomes available. The transformer model, which is gigantic, yeah, in its part. It's this was $100,000 in training cost with Google, and it was highly rebated only. So this is a very valuable model, yeah? But we are making this available. We are talking to OECD, we're talking to NIH to make this just freely available that anybody can use it. But the agentic AI around this, which has 1,500 agents at the moment, which can be orchestrated and are constantly self-optimizing, that's a different beast. Yeah? But that's making a lot of sense of these. Yeah.
Thank you.
Okay. Thank you very much for having me.